Everybody, welcome back to another AP Biology video. In this video, we're going to cover AP Biology topic 5.3, which is on the topic of Mendelian genetics. We're going to talk about the types of inheritance that primarily operate under the law of dominance as established by Gregor Mendel, who we're going to talk about here on the next slide. So if you guys are ready, let's get into it. Now, you may be familiar with the man that is Gregor Mendel. He was an 18th century monk who studied genetics using pea plants that were grown in the monastery garden. So he used these pea plants as model organisms for a couple of different reasons, but they grew quite fast and they were easily able to control in terms of what mated with what. But it was in this investigation that he was really looking at characters, or what we would call the inherited features in different individuals. And he looked at 7 different distinct characters which had different traits associated with them. Here we're going to use the term traits to describe the different variations in character. So I'm going to use primarily today looking at flower color, which he took note of. You had 2 different traits that were associated with the flower color character, those being purple or violet and white. So let's talk a little bit about what is a gene. So a gene is going to be this segment or sequence of nucleotides. Remember, nucleotides are the monomer of our nucleic acids. So this includes things like DNA and RNA, which we find in all of our living things. They have some sort of genetic code that decides how their function and structure are really put together. So it is these sequences of nucleotides, the segment of our DNA, that determines what the character or trait of our organism is going to be. Now, when it comes to genes, genes are located at a specific point along our chromosome, and we are going to call that the locus. So if we were to look at 2 homologous chromosomes, the gene for the same character would be located at the same locus between the 2. Now, we do not just have one gene, otherwise everyone would look the same. So what we do have is different versions of a specific gene that we call alleles. And alleles in Mendelian genetics come in 2 different flavors. We have dominant alleles, which are expressed when they're present, and we have recessive alleles, which are masked if in the presence of a dominant allele, and only expressed if we do not have a dominant present. So when we take into consideration that we have 2 different alleles, that means that we can combine them in a couple of different ways. So we can put 2 dominants together, we can put 2 recessives together, and we can put a dominant with a recessive. So the combination of the alleles is what's going to determine which character is expressed. So here we have a couple of different flower colors, the purple in this case being our dominant trait because it is expressed when that allele is present, and we only get that white flower color if the dominant is not present. So it's this outcome, these physical traits that are expressed, that we call the phenotype. I like to associate the term phenotype with physical because that reminds me that these are what we see or what we're able to measure coming from an organism. It's not the code itself, it's the programming. So in these phenotypes, we have a couple of different colors, and those are going to be determined by what we call the genotype, which is the term that we give to the combination of alleles possessed by an individual. And as you have seen, we have a couple of different ways that we can organize these alleles, different combinations, and we give specific names to these. So if we have two of the same allele, we term those homozygous. So homo comes from the term meaning same. So if we have two identical alleles, we are homozygous. We can be homozygous dominant. We can be homozygous recessive. But as long as we have two of the same alleles, we are homozygous. If we have two different alleles, say one dominant and one recessive, we term that individual heterozygous. Hetero meaning different. So as a culmination of all of his research, Mendel was able to develop a few important fundamental laws of genetics. So the principle of dominance, which we've already talked about, is this idea that we have one allele that may be expressed whenever it's present, and it may mask the presence of another allele. The other two fundamental laws that we're going to look at today echo back to what we saw during meiosis. So we have this segregation, the idea that our alleles are separated when our gametes are formed. To segregate means to separate something. So each of our gametes is going to inherit a different chromosome from our parent cell. So we have this splitting off of our genes, and then those may be sorted independently, echoing back to the independent assortment that we saw during metaphase 1 into the different gametes. So we get all of these different possible combinations. And so if you have a specific gene for eye color, you're not necessarily going to have a specific gene for hair color. They are separate genes, and therefore they are inherited independently of each other. And one of the ways that we can really visualize these laws is through the use of Punnett squares. Now, you may be familiar with Punnett squares from, say, a middle school life science class or even an earlier biology class if you took one. But they are a really good way for us to really break down some genetics problems. So I'm going to show off two of our main types of Punnett squares. We're going to do a monohybrid and a dihybrid. Those terms referring to how many genes we are looking at. If we're dealing with a monohybrid cross, we are going to be looking at a single gene. Mono means one. So I've got a problem set up here where we have a pea plant that is heterozygous for purple petals. So that means that they have a dominant and a recessive. Our dominant here is represented with a capital letter, whereas our recessive is represented with a lowercase letter. Now we've crossed this heterozygous pea plant with another heterozygous pea plant. So both of them express purple petals, but do have that recessive character still in their genotype. So the first thing that we want to do as far as setting up a monohybrid Punnett square is to take our parents and place each of their alleles on a separate box. So here we have the first parent, and they're going to go on top, and then we're going to put another parent on the side. So their alleles are going to be in separate boxes. There's our second parent. And from here, we are going to show which possible combinations our offspring can inherit. So we're going to pull that first parent's alleles down into that column, both alleles inhabiting a separate column. And then for our second parent, we're going to pull their alleles across. So we'll go and fill in that row with each of those. And so from here, we can see what the potential genotype combinations of our offspring could be, and we can then determine which phenotypes should be expressed in which ratios. So here we have two dominant traits. That's a homozygous dominant, and that is 1/4 of our potential offspring. We have two that are heterozygous, and so that gives us a 50% chance of having a heterozygous offspring. And we have one that is homozygous recessive with those two recessive characters. From these traits, we know, based on the law of dominance, that we will have, if we have a present dominant trait, that we are going to express that dominant phenotype. So we have altogether a 75% chance of having purple offspring, and then only our homozygous recessive is going to translate to a white recessive phenotype. Now, monohybrid crosses tend not to give too much trouble. They're pretty straightforward, and once you get the hang of them, they're not too bad. What gets students a little bit more confused, or what maybe worries them more, are our dihybrid crosses. Because our dihybrid crosses take into consideration two separate characters. So we have two separate genes here that we're looking at. And so some of the trouble that students have is determining what the potential gametes of these individuals could be. So that's where we're going to start. In this case, we have two pea plants. They are heterozygous for purple flowers and green pea seeds. And so that gives us two separate heterozygous genotypes. Now, to take this number 4 and turn it into the potential gametes, we're going to use a process called foiling, which you may be familiar with from math class. So in foiling, and that is a mnemonic device, so we can go first, so that gives us a gamete with both of our dominant characters. We can go outside. Let me spell foil correctly. Outside, so we're going to get a dominant H and a recessive G. Now it's inside, so we're going to get that recessive H and that dominant G. And then last, so all together that spells out foiled, so we get both of those recessive characters. This foiling process exhibits both our law of segregation and our law of independent assortment. So each of our gametes is getting one of our genes, and we have independent assortment of those two genes together. So I've already gone ahead and set up the traits as associated with each of those gametes for parent 1 and parent 2. And so just like before, we're going to pull the traits down for each of our potential gametes, and we're going to pull them across for our other parent. So we want to make sure that all of our offspring end up with the same number of genes as was exhibited in our parent. So each of our offspring should end up with 4 genes in this case, echoing those of the parents who had 4 genes. Now, what is a little bit more fun when it comes to dihybrid crosses is really identifying the proportions of our genotypes and our phenotypes. So I went a little crazy with the colors here, so I apologize if it's harder to read. But we have 1, 2, 1, 2, 4, 2, 1, 2, 1. Now, what we can look at here when we count up all of the potential genotype combinations is we can determine individual phenotypes. For example, we'll have 12 purple plants, 4 white plants, 12 green plants, and 4 yellow plants. But what we're really more interested in is the proportion that we could potentially see if we had the combinations of these different phenotypes. And this gives us a very important ratio, and it'll be more important when we get into the statistics. So it is one to keep in mind. But if we cross 2 plants that are heterozygous for a dihybrid cross, we would get a ratio of 9 to 3 to 3 to 1. So we would see most of our individuals exhibiting those dominant characteristics, and then the proportion would decrease as we went from there. The time that you have on the AP exam, sometimes it's a little much to ask students to draw out a full dihybrid cross. You will see that it takes a minute unless you're really fast. So one of the things that we like to do is to introduce the concept of probability. Much like with a coin flip, because the genetic crosses represent independent events, we can use probability rules to determine potential outcomes. So if 2 events are equally likely, the outcome does not affect each other. So we get 2 rules that we can associate with this. One of these being the rule of multiplication, which we can also term the product rule. And this states that the probability of 2 independent events occurring together, so we've got both events occurring at the same time or in sequence. This is going to be calculated by multiplying each of the chances of the events occurring alone together. So we call this the AND rule because we have both cases together. And this is echoed in the equation that we use for this, which states the probability of A and B occurring is equal to the probability of A times the probability of B. So let's look at a practice problem with this one. Let's say that the genotype of 2 individuals in a dihybrid cross is dominant A, recessive A, dominant B, recessive B. So we've got heterozygous individuals for both traits. And we want to know what the probability is that our offspring would be homozygous recessive for A and heterozygous for B. So what we're going to do is we're going to first identify our 2 parents. So we've got 2 heterozygous individuals for both traits. And we want to figure out what the probability of homozygous recessive and heterozygous would be. So we would want to see the probability of potentially creating an offspring that has a lowercase a and a capital B, and the probability of having 2 recessive traits for those 2 characters. So we would have a 1/4 probability times a 1/4 probability. That would give us in total a probability of 1/16 for this final offspring. And this is not the only rule that we can use. We also have what is known as the rule of addition, or our sum rule. And this is looking at events occurring independently of each other where we could have either or. So the probability that one independent event or a second independent event will occur independently is calculated by adding their probabilities together. So this is our OR rule. And we can see that in its equation, we have the probability of A or B occurring is the probability of A plus the probability of B. So let's look at another practice problem. So in this problem, we have a genotype of two individuals and a monohybrid cross is heterozygous. What is the probability that our offspring would be heterozygous? So we would establish first our parents. We have two heterozygous individuals. We want to figure out what is our probability of having heterozygous offspring that have either of those two associations. So we could have the dominant first from one parent and the recessive from the other, or vice versa. So that's where our two probabilities are coming in. So this would also give us a 1/4 probability that we would get each of these individual outcomes. So adding them together gives us a probability of 1/2, which is the same thing that we saw in our monohybrid cross. Now, while I've got you here talking about traits, it is important to note that our characters, our genes, are associated with different chromosomes. So you probably know that humans have 46 individual chromosomes and 23 pairs, so different genes show up on different chromosomes. We can look at the organization of our chromosomes by taking a look at what's called a karyotype. A karyotype is essentially a model that shows all of the chromosome pairs in an individual. This, for example, is a haploid individual because we have one set of each of our chromosomes. There are karyotypes out there for diploid individuals as well, but we can differentiate between our chromosomes based on what types of genes they carry. So first off, we have autosomes, and autosomes are going to carry our traits that are not representing our sexual characteristics. So that makes up the primary number of our chromosomes. 1 through 22 in humans are our autosomes. And then we have sex chromosomes. Set 23 are our sex chromosomes, and these carry genes that are specifically coding for sexual characteristics. So all of the crosses that we've looked at so far are what we would consider to be autosome crosses, because we're not looking specifically at sexual characteristics, or we're not looking at traits that have been associated with sex chromosomes. So we do have cases where we have genes that are specifically inherited along our sex chromosomes, and the ones that we're going to be really focused on in AP biology are those associated with our X chromosomes. So if you recall from middle school life science that you have females that inherit X chromosomes from both parents, so they are homozygous for that X chromosome, and males are what we would term hemizygous because they have two different chromosomes, not just two different alleles. They only get their X chromosome from their mother. So when we have genes that are carried along the X chromosome, we can represent them in a slightly different way. So we always get the X, but we have a little superscript there to tell us if our X trait is dominant, or maybe giving a superscript lowercase number to show that we are recessive. Now, beyond this, the law of dominance still operates the same way. So we can have a homozygous X trait, we can have a heterozygous X trait, and we're going to look in the next video at how some of these different characteristics can be inherited across generations, and to allow us to look at how different disorders are prevalent throughout. So I will catch you in the next part of our 5.1 video on the topic of pedigrees. Have a great day and take care.